In our screencast today, we're going to talk about the plasma membrane. The plasma membrane is pretty awesome, and let's find out why. So the plasma membrane is the boundary that separates the cell from the living cell from all its surroundings. And it is basically the reason that life exists. Now we can establish an entirely stable and different internal conditions in the cell than external conditions. The plasma membrane exhibits selective permeability, allowing some substances to cross the membrane and other substances stay out. And that's, again, how it can allow exchange, so the cell gets what it needs, gets rid of wastes, but also make sure that things that are harmful to the cell or not good for the cell will not get in. Selective permeability, key term. Okay, pardon me. So membranes are fluid mosaics, we can get to that in a minute, of lipids and proteins. So phospholipids are the most abundant lipid in the plasma membrane, by far the most abundant membrane component, period. Phospholipids are amphipathic, which means they have both hydrophobic and hydrophilic regions. And the fluid mosaic model basically states that the membrane is more like a fluid, so like a liquid, than it is like a solid. So it's constantly flowing and moving. Think of it as like an ocean of lipid. And then it has this mosaic of proteins, had all these like little protein sort of rafts floating around in the ocean of the membrane. So if we look at some models of membrane, they contain, when you chemically analyze them, they contain proteins and lipids. And because of the amphipathic nature of the phospholipid, scientists who study the membrane figured out that it must be a bilayer, 2 layers of phospholipids. Okay, and here we see our 2 layers. There's 1 layer and there's 2 layers. Okay, and again, because the hydrophilic head likes water, it orients itself to face the water on the, let's say this is the inside of the cell and this is the outside of the cell. And then this orients the hydrophobic tails towards each other, and they can kind of hide. But it's this hydrophobic region that really is the magical component. It creates a barrier that a lot of molecules cannot cross, at least not without assistance. So that's kind of the key component to the structure of the membrane. In 72, Singer and Nicholson, you don't need to know them, proposed that the membrane is a mosaic within the bilayer, and then the hydrophilic regions are exposed to water. And this is basically kind of just confirming what we know. And so here is our phospholipid bilayer, the hydrophilic head region, hydrophobic tail region, and then there are all of these proteins kind of embedded and swimming and floating around in the membrane. And these proteins are also amphipathic. Okay, if they're going to be in the membrane, the region that is in the hydrophobic tail region has to be hydrophobic, right? Like dissolves like. Can't fit in my phobic. Okay, and then the regions outside of the membrane that face the water or the aqueous solution within and external to the cell have to be hydrophilic. Okay, they have to be, again, like dissolves like. Okay, there are lots and lots of things associated with the membrane, present in the membrane. There's glycolipids, which are lipids attached to carbohydrate. There's glycoproteins, which are carbohydrates attached to protein. Okay, there are proteins that span the membrane. There are proteins that are either on one side or the other of the membrane. Okay, the membrane's anchored to this out here is called the ECM, or the extracellular matrix. And it basically kind of holds all of our cells together. It's attached on the inside by the cytoskeleton and helps the cell to keep its shape. So it really is a very complex structure. We'll get into more detail there. So let's focus a little bit on the fluidity of membranes. Again, membranes, it's hard to think of this thing that encloses our cells as a liquid, because like, how can that be a barrier? But it really is more like a liquid than it is like a solid. There's a lot of movement within the membrane. Okay, so phospholipids can move within the membrane. Most of the time they just drift laterally. They really don't kind of flip-flop within the layers. So let's see how that looks. So again, here you get movement, back and forth, side to side within the layers. Flip-flopping across the membrane is pretty rare. Okay, lateral movement occurs very often, 10 to the 7th times per second. That's a lot of movement. But the flip-flop is very rare. Comparatively, only about one. Also contributing to the fluidity of the membrane is temperature, right? So temperature is a measure of average kinetic energy. So things move faster when they're warmer and slower when they're cool. So again, this can also affect the fluidity of the membrane. So if you cool off, membranes will go from a fluid state to a more solid state. Okay, the temperature, and this is actually not helpful. So the temperature at which membrane solidifies depends on the lipids. So if you have, again, unsaturated fats, they have those kinks in the tails, and they space themselves farther apart, pardon me, because they can't get closer together, so they will stay more fluid. If you have saturated fatty acids, they have the straighter tails. They can pack in more tightly, and it'll always be more solid. Okay, membranes must be fluid to work properly. Okay, they're usually about as fluid as like olive oil. So, picture like your membrane is like olive oil. Things that can help the fluidity of the membrane, other lipids, the steroid cholesterol, actually has different effects. If you warm it up, it actually restrains the movement of the phospholipids and makes it more solid. But where it does help is at cooler temperatures, it prevents tight packing and it maintains fluidity. So cholesterol, good in the cool, not so good in the warm. And so here, if you look, unsaturated fatty acids, okay, see the kinks hold the lipids more farther apart, so they can't solidify, and that will help retain fluidity. If you have saturated fatty acids, they get very tightly packed, they get more viscous, more solid. And then cholesterol, you see here's our cholesterol molecules. Again, at high temperatures, they actually help them to pack, but at lower temperatures, it can hold it further apart and help to maintain fluidity. Okay, so again, evolution and differences of membrane lipid composition. If you think about it, organisms live in lots of different climates and lots of different temperatures. So you can look at the plasma membranes of animals like, polar bears or other things that live in very, very cold temperatures, and they have a different composition than animals that live in the tropics or plants that live in the tropics because of the difference in temperatures. So it's really kind of cool. So they are adaptations. So the lipid component of your membranes are adaptations to environmental conditions. Okay. And also, if you can change, there are some animals that live and plants that live in areas of extreme temperature changes. Okay. So if temperatures vary greatly, they have evolved the ability to change the lipid composition of their membranes, which is really kind of cool. So there's kind of part of our evolution component for this chapter. Oh, and that's the end. We will continue on with the plasma membrane in the next screencast.